Right. We're in chapter 3, cells, and we're into movement of substances across the cell membrane, through the cell membrane. And first, we're going to talk about why it's important to even talk about what's going across the cell membrane. Okay. Cells, first of all, are really small, right? We all learn that. You can't see cells unless you're looking at it in a microscope. And why is that, though? Why don't we have cells that are larger than microscopic, really? So here's why. What limits the size is that fact that cells rely on their cell membrane for absorbing appropriate amounts of nutrition or nutrients and getting rid of waste. So we say excreting waste. All right. What happens if the cell is larger, then that surface area where the cell membrane is at, we're talking about a sphere here, the surface area of the cell will get too small if that ratio of surface area to volume. So proportionally, that ratio of surface area to volume has to stay high enough to maintain the contents of the cell. But if the cell's too large, that ratio gets too low for the cell to survive. Okay, and I'm going to do a little tiny bit of math, very tiny bit, just to kind of demonstrate that. Okay, here we go. Just do it up here. Surface area equation to calculate it is of a sphere is 4 pi r squared. Okay, and we're talking about a sphere here. It's a really bad sphere. Okay, for volume, it's 4/3 pi r cubed. So if you do the ratio, you put one on top of the other, surface area to volume. So we're going to put surface area on the top. Okay. Remember, we factor out everything that's on the top or divides out. So 4's divide out, pi divides out, 2 of the radius divide out because you have 2 on top, and then you're left with one radius on bottom, and then you're left with this 3 that's on the denominator of the denominator. Remember, you want to try to move it by dividing it out from that position. So that divides out. I'm not supposed to say canceled out. We get 3 over radius. That's what your number is of the ratio of surface area to volume if you know the radius. So as the radius gets bigger, since it's on the denominator, this fraction gets smaller. And proportionally, it just gets too small to sustain all the chemistry that's happening on in the cell, all the cell's metabolism. It doesn't get enough nutrients, can't get rid of all the waste. So yeah, cells don't have like a circulatory system. They just use a cell membrane. So we're going to talk about movement across the phospholipid bilayer membrane, right? That's what our membrane's made out of, is those phospholipids. And the deal is, remember we have the polar heads for the phospholipid, right? They're negatively charged. I won't draw them all. And then the nonpolar legs, and they kind of group together with the opposite layer. So that's what the bilayer is. Okay, so that's just how we kind of sketch phospholipids. Nonpolar in the middle. You probably sketch it a lot better. And some things can wander. Okay, things can wander by themselves, maybe passively, and we're going to talk about passive transport where no energy is required if they are small, uncharged molecules or particles, or atoms, molecules. They can pass through the membrane. That really limits what can pass through on their own. So if we're talking about passive transport, it's probably like water, H2O, sorry, yeah, oxygen, carbon dioxide, which is good because you want the cell to be able to just absorb as much oxygen as it needs. So the oxygen will diffuse because the cell oxygen level is getting a little bit lower, and hopefully the blood oxygen level is high enough. And vice versa with the CO2. But no salts, no salts. They can't. Okay, but I just want to show you the situation in my class where you can affect things that go on in the cell without having molecules actually go across the cell membrane. We're gonna, as we probably can see here. Oh gosh, I could mention steroids. All right, steroid hormones. Sorry, that's separate. Can pass through, and then they can actually act on the DNA kind of directly. But a lot of other molecules that are signaling molecules, they might actually associate with a protein receptor or interact with a protein receptor. So I'll just draw that just for grins because that protein receptor can span an entire membrane. Remember, it's embedded in this phospholipid bilayer membrane. And they've got a ligand that cannot cross the cell membrane, but it could bind to a protein that is designed for it. A ligand is just a fancy word to say molecule that's going to bind and do something, I guess. So ligand that cannot cross the cell membrane can bind to a protein that's a receptor. It's made for it. And it causes conformational changes. Remember, proteins have those hydrogen bonds, but when something binds, it's going to kind of disrupt it, possibly. And that's what can cause a cascade of signaling. Okay, so we like a conformational change on one end and can actually affect the other end. Okay, maybe so a binding on one end can affect and make a conformational change on the other. Okay, so that could be like number one, number two on the cell, inside the cell. Maybe the signal gets initiated. And it's a cascade, usually, of a chain reaction. And there's possibly other regulation points. And if you're into molecular biology, you're going to study that. And it's really fascinating. Cyclic AMP is usually oftentimes used for a cascade event. So that's how you can affect events that go on in the cell without even having hormones enter the cell. Some hormones can, like steroids, but molecules we need in a cell, like glucose and nutrients and stuff like that, they're going to need other things to travel across the cell surface, like proteins. But first, we're going to talk about passive transport. No energy required, passive transport. And our favorite is diffusion, right? Say number one, diffusion. And that's just your normal molecules moving from an area of high concentration. So these are all supposed to be dots of like some sort of molecule. To areas of low concentration. And that's with the concentration gradient. Okay, so there's like a gradient, a difference. So we'll say that it moves with the concentration gradient. Okay. If there's other molecules in here, it doesn't really count. It's just the molecules are like, oh wow, you're a lot like me. I'm going to go where there's a lot of less of me. All right. So even if you have other molecules in here, those molecules still want to spread out. It's like, you have air everywhere, you have atmosphere everywhere. But if somebody like lets loose a fart in one side of the room, it's going to diffuse to the whole room, right? Regardless. If it's a good one, people will be able to tell. If it's not such a good one, It's just going to blend in with the smell of like, dinner or something. So yeah, hopefully it's a good one. Facilitated diffusion. So that was passive diffusion or simple diffusion. Sometimes we'll call it simple diffusion. Facilitated diffusion is the same idea, only they're going to need the help of a selective protein. So molecules diffuse through a membrane with the concentration gradient, or we could also say down the concentration gradient. So that too. But we need this channel protein, and how we usually draw it is your phospholipid bilayer membrane. I usually draw it with like a hourglass, and it's a channel, and it's exclusive to only certain molecules, probably smaller molecules, but that's okay. Small molecules that are attracted and in a certain way to whatever amino acid side chains are in this protein, the center of this hollow protein. So it's a channel or gated protein. A channel could be open all the time. Well, I'll just write over that. Or gated, which means could be opened or closed by another factor. Okay. So, for example, we could have sodium, maybe low concentration of sodium here in the cell, and you have high concentration out. Oh, I think it's usually the opposite situation. That's okay. And normally, the concentration gradient is going to say, okay, it's going to travel down from area of high concentration to low concentration gradient. So it travels with or down the concentration gradient. And because sodium is charged, it can't get through the membrane by itself. It needs this channel. So this channel protein has amino acids that attract certain molecules. Okay, and usually then, yeah, it'll only allow those small molecules to kind of pass through. All right. If it's gated, then it's going to be open and closed by factors. Okay, so gated channel is going to need a cofactor, usually, or conditions. Certain conditions will cause them to open or close. Like chlorine channels are open all the time, usually for balancing. Glucose channels, I think they need insulin to open them. Like you don't remember that your cells don't actually absorb glucose. It's too polar of a molecule large, and so it needs that insulin cofactor. So maybe chlorine channels are open, usually in cells, especially in like muscle cells. For example. So those are types of facilitated diffusion. And then the third type of passive transport, remember we're talking passive, no energy required, is osmosis. And the reason why is because water can wander through membranes. By the way, cholesterol slows this down a little bit. Yeah. But that's okay. It still water wanders. There you go. And so we're going to talk about what wandering. It gets its own special name, osmosis, because osmosis parted the waters or moved the waters, I guess. So yeah, hypertonic solutions, hypotonic and isotonic. So I want you to draw 3 cylinders here with space in between. All right, mine are not consistent. They're supposed to be the exact same size. Okay, what's important is You're going to draw a little circle to represent a cell. Try to make the cell the same size. Oh gosh, not even close. That's okay. Hypertonic solution hyper means above. So it means above level of saltiness than the cell, or above the saltiness of the cell. So the solution itself, well, we got to put some solution here. Sorry about that. There we go. Let's say the cell is only 20% salt and 80% water. Okay, doesn't really matter, by the way, what kind of salt. Water just counts it as like stuff other than itself. It's 20% stuff other than itself and 80% water. So if water doesn't care if it's potassium chloride or sodium chloride or glucose or whatever. Okay. And then the liquid around it is, let's say it's 35% salt. So it's saltier, or dissolved solids, and 65% water. So the water is saltier if it's hypertonic to the cell. We're talking about the solution being hypertonic, salty water. Okay, so what happens here? Yeah, the water wants to travel from areas of high concentration to low concentration, and it can, and it does. So water is going to, yep, you guessed it, move out of the cell. And what happens is the cell shrivels, right? So if the cell shrivels, he's not super happy. It's not a really great situation. That's kind of an exaggeration, but what's important to note that is, even though the cell might survive, especially if eventually you can get back into some less hypertonic solution, but if you drink salt water, it's going to dehydrate your cells. That's why you can't drink salt water in the ocean. You need fresher water. You're already eating salts and other dissolved particles in your food. You don't need too much more. So yeah, it's not good, but here's a worse situation. All right. So let's say in the hypotonic, hypo means low. So it's below the salty level of the cell. Let's switch the situation. Let's say that the cell is 35% salt stuff and 65% water. And let's say that the water around it, the solution around it, is 20% salty stuff and 80% water. Okay. So where's the water going to move? It's going to move where there's more water to where there's less. So water is going to fill that cell. But as you can see, there's a lot more water than cell. And even if it was a microscopic cell, it would be even more so. That cell does not have the capacity to take on all of that water. So the result is that the cell is going to swell. And if it's salty, or if the cell is salty enough, or if the water is hypotonic enough, the cell is going to burst. That cell membrane cannot stay together. And the cell dies because it ceases to become a cell. The cell has to be able to stay together. So the cell bursts, that's bad, and dies, right? All right, so again, hypertonic solution, water moves out of the cell. Okay. I like to think of it as the word, like a word picture, hypertonic and hypotonic, to kind of remember what happens if the solution is hypertonic. Then I usually think of the R as the shriveled cell, and hypotonic, the O is the swelling cell. Okay, so if you're ever in a situation, in a test where you're like, you're just kind of nervous and it's hard to think about, you can just think about word pictures like that. The last situation is isotonic. Iso kind of means same, right? So that the salt content is the same inside and out. Let's say they're both 35% salt. Oh, where'd my red go? Therefore, they're both 65% water. 35% salt and other dissolved stuff, 65% water. Okay. And what happens? Yeah, the cell stays the same shape, if you can draw it the same shape. But Yeah, where does the water move then, or does it not move? By the way, the cell's pretty happy about that. That's why it's important to have balanced, osmotic balanced um, solutions around our cells and in like our eyes and our membranes where the cells are more vulnerable. They need to have a certain level of saltiness. Both, by the way, happens. Yeah, the water wanders no matter what. So it's gonna move no matter what. But what happens is it moves out of the cell and into the cell at the same rate. So the cell stays the same shape and size and doesn't die. So water moves always, but in this case, it moves both in and out at the same rate. Okay, got it in there. All right, so hopefully that makes sense to you. You get that in your notes, and now we're going to talk about active transport. That's the type of transport that's going to require energy, and it's usually ATP, not 100% of the time. But what's another defining feature? Active transport molecules are moving against, or they could move against the concentration gradient instead of with the concentration gradient. So we're talking about water moving or substances moving from area of low concentration to an area of high concentration. Coincidentally, water, there's no pump for water. Like you can move water, I guess, or move salt out of a cell and water will follow. And that's how you sweat. And that's why your sweat is salty. It has to be. Otherwise, we wouldn't be able to sweat. We wouldn't be able to lose water. Okay, so that's a defining feature, moving from possibly from low concentration to high concentration. And so it's against the concentration gradient, or yeah, and requiring energy, usually ATP. All right. And then usually we have carrier molecules involved. Yeah, because we need stuff to happen. So we're going to need some proteins here. All right. So here's an idea. I think they used to call this bulk transport. We don't really use that, I guess, term anymore. Or bulk transport doesn't actually have to do with exocytosis of this type, I guess. Okay, so maybe save that for later. Exocytosis, though, it means exiting the cell. So I'm going to say, yeah, same kind of idea of the channel protein, only this one's just a little more high tech. And the conformational change is going to like literally pull the molecule across when it does its conformation. It's going to do a conformational change with energy, with ATP. Okay, so let's say we have a situation where we have high concentration of sodium and a low concentration of sodium, and it's still going to pump one at a time sodium molecules across the cell membrane with the use of a carrier protein. And it kind of is like a pump. And it usually has ATP, and it may be a cofactor to tell it to do that. To like, okay, it's time to do that. Maybe. A cofactor. In the case of your muscles, it's just going to be constantly like, as soon as the cell depolarizes, it's going to try to, these pumps, carrier protein pumps, are going to try to repolarize the cell. So, yeah, that happens. We'll study that a little bit later. Okay, so ATP gets used, and that's cool. All right. I'm going to do a side nod to filtration. Filtration is how your kidneys get rid of like most of the majority of your non-cellular material in their blood so that it can try to grab back what it needs, so it will be able to filter it. So it pushes through plasma and dissolved molecules and small particles with the help of blood pressure. And of course, so blood pressure is energy. We had to put energy into the system. If your blood pressure is not high enough, your kidneys are not going to be able to do this effectively. So they do use blood pressure, but the molecules go from high pressure to low pressure, of course, but no ATP required. So that's one situation that I know of that in this filtration that it's going to require no ATP, but it does require high pressure to low pressure. So it requires blood pressure. Likewise, if your blood pressure is too high, it's going to blow out your little tubules and capillaries and stuff. So that's not good either, your kidneys. The next type of transport is taking things into the cell. Yeah, so that might be kind of, I guess you could, yeah, you can pump things into the cell using this receptor mediated endocytosis. I guess that's fancier. Or you can take things and have it exit the cell too. So I guess you could do that for both. Okay, but for endocytosis, the classics are pinocytosis and phagocytosis. Yeah. So to eat is to phage, or we would pronounce it phagocytosis, and to drink is, or a drink is like pinois. I guess this is cell eating and cell drinking. So liquids are literally taken in and the cell engulfs it. This is how your kidneys sort of like take back what it thinks it needs. It's not super efficient, honestly. Human kidneys, I hear, are just not actually that efficient compared to other species. But the cell will sense, it maybe has like little receptors that sense that there's liquid in here and it just wants to engulf the whole dang thing. Okay, so receptors sense the presence of liquid and they just kind of, invaginate, make a, they're going to make a vesicle. So the cell membrane surrounds the liquid. All right. And then ATP is also like all the time. Yeah, you're using a lot of ATP to do this. So there we go. And creates a vesicle. All right. So that's one thing. Remember ATP involved. And then ATP is uncharged to ADP. Also, that might be happening around here too, but that's okay. So yeah, when the cell is invaginating, then it's like it's moving. It's got to use ATP for that. Super complicated process. It's pretty awesome. Biochemistry, if you want to study that, it's great. It's fun. Phagocytosis, cell eating. It's just basically the same idea, but with solids. And the solids could be dissolved or they could be just floating. But if it senses it, it's going to form a vesicle around it and pull it into it. So I'm just going to say same idea, but with solids, and it does require ATP. The next type of transport, the receptor-mediated endocytosis is, so you could have receptor-mediated exocytosis is what I was trying to say. Maybe I'll write that down here too. Okay. But we're saying endocytosis into the cell, it's just a selective protein which binds and they're saying it's a 1-to-1. It's not just like, this looks good and kind of blindly making a vesicle around it. It's a taking the binding to the molecule and taking it into the cell specifically. So it's more specific, requires more ATP, and yeah, I can be against the concentration gradient then. So if your cell needs it, it's going to pull it in. A lot of times your intestines are doing this. Let's draw a little cell here with these receptor proteins that span the cell membrane. And they're like, this is what I like. and they're going to bind to it, and they're going to kind of congregate, and they're going to create a vesicle as well. I suppose you could have it pulling the protein across a channel, but you need a channel for that, like we talked about before. Still, ATP is required for these, but you get a lot of stuff specifically in that you need it. All right, where does energy come from? ATP, right? Which comes from cellular respiration. Because we all know you could do glycolysis, but it's not going to give you as much ATP. So really, the most bulk amount comes from cellular respiration. All right, very good. Thank you, ladies and gentlemen, for following along on this journey of cell transport, and hopefully that made sense to you. I hope you got a chance to try to label the cell, maybe print out these notes, and there she goes. Yeah, these are the parts of the cell that we're going to be studying. Also, you'll want to study the functions. All right, so that says cytoplasm, by the way. All right, could be cytosol or cytoplasm. And then what parts of the cell is most like what? Well, that's kind of related to the definition. So hopefully you kind of got those notes from the last video, well, in class, or kind of we went over that, but I don't know if we had time to go over all of it. So anyway, let me know if you have questions, and hopefully you have a nice weekend. And yeah, we'll see you soon.